Hello everyone, welcome back to another class on regeneration biology. So in today's class, what we will learn is a brief outline about different tissues regeneration, and we start with fin regeneration. So, and the word fin itself make you understand that we are talking about a fish, and that species is zebrafish. And we will come back to the regeneration of fin in detail in a later class, but first we need to introduce the species and also what is the basic things we need to know about regeneration. So this is zebrafish. Many of you would have seen in the pet shop and which is very prevalent in Indian aquarium pet shops, and also it is native of India. And zebrafish is a very interesting interesting species of fish, and it belongs to the genus Danio and comes under the family Cyprinidae. That is a bony fish group. It's also called teleost fishes. So zebrafish is native to the streams of southeastern Himalayan region and is found in parts of India, Pakistan, Bangladesh, Nepal, and Burma. And the species arose in Ganges, the river Ganges, and in eastern India, and is commonly inhabits streams, canals, ditches, ponds, and also it can also live in stagnant water bodies and even in rice paddy fields, etc. Means its ability to grow in a diverse water habitat is known. It's a freshwater fish, and hence it can be easily manured in laboratory condition. The zebrafish is named for 5 uniform pigmented horizontal blue stripes on its body, which is reminiscent of the animal zebra and which extend to the end of the caudal fin. It walks into the caudal fin from the part of the body, and it shapes its fusiform and laterally compressed, and its body is fusiform laterally compressed and its mouth slightly directing upwards. So male zebrafish is a torpedo shape with golden stripes, slight yellowish golden stripes between the blue stripes, whereas the females have larger whitish belly, which is because of the presence of the ovary and the yolk, whitish belly, and silver stripes instead of gold. So you can easily identify slightly color variation is there, blue versus yellow, and also the body shape also. It will have a belly in the case of female. Adult females exhibit a small genital papilla in front of the anal fin origin. So if you use a dissection microscope, you can identify the female through its genital papilla. So there are ways of easily identifying this male or female gender in zebrafish. It's an excellent regeneration model due to various features and factors associated with this animal. 2 features which make the teleost fish, zebrafish, a powerful complementary model system to study organ regeneration. And we all know to study organ regeneration, we have to get into the gene expression or different types of genes, genomes, etc. And zebrafish share almost 70% homology to that of human, and quite a lot of the genes have extensive genomic synteny. That means the order in which the genes are arranged. So this is quite beneficial because the order of genes also play a major role in their expression pattern. First, they are highly regenerative, equipped to regrow amputated fins, injured retinae, transected optic nerves, spinal cords, and resected heart muscles, scales, etc. Second, unlike the salamanders, which is the amphibian, they are responsive to both forward and reverse genetic approaches. Salamanders, these forward and reverse genetic approaches are limited because they cannot give as many number of eggs, and they may not breed throughout the year as efficiently as that of zebrafish. And the genomes of quite a lot of salamanders are very lately identified, and some of them are sequenced recently. So the forward and genetic screen approaches also depends on the size of their eggs, whether they are amenable for microinjection or maneuverability. Some of them may not be very transparent in their embryonic development. Zebrafish has got excellent transparency in its embryonic development. Hence, these are all make it very easy model organism to monitor. And when it comes to regeneration, it is as good as that of amphibians. As is customary with the genetic model system, various community resources are available for gene discovery and molecular characterization of zebrafish. This is an important feature, that if you are studying a species, the availability of resources or reagents across the research groups or research fraternity is very important. If you are starting a new species and no resources are available, you have to identify, discover those features and then study. It's almost like you have to make the road and ride on it. That can be very cumbersome when it comes to doing a effective or easier research, including mutagenesis screens, transgenesis, microarrays, developmental markers, and genome sequencing informations are must if you want to study on a species. Zebrafish fulfills all these criteria. And zebrafish fin regeneration is very historically important, because it is quite compelling to know that almost 250 years ago itself, people know that fish can regenerate. Like even we have seen in the planaria, when we studied planaria, many scientists have reported the regenerative capacity of planaria. Same way zebrafish regenerative capacity also people knew it. But it is a sad fact that despite having knowledge about the regenerative ability, means one of the earliest research area or research topic that has emerged for the scientist, it still remains incomplete. That means although we know a lot about the regeneration mechanism, we are far from eliciting these regenerative tactics or skills of lower vertebrates in higher vertebrates such as mammals. So that is some some area that need to be fulfilled. The ability of teleost to regenerate amputated fins was first reported in 1786 in the pectoral fins of goldfish by Bossonet, the scientist's name. Although Thomas Hunt Morgan, the famous scientist who have done a lot of work on the mutagenesis or mutations, etc., was fascinated by fin regeneration at the turn of the beginning of the 20th century. Fin regeneration took nearly an additional century to reach the genetic era. That time people don't know too much about the genes, genotypes, genomes, etc. They know it regenerates, that's it. Even cell biology was in its infancy during then. In 1995, Johnson and Weston described a screen for mutations that disrupt the regeneration of tail fins in adult zebrafish. That is almost 30 years ago. Later, a handful of mutations that inhibited fin regenerations, and that could be localized to specific molecular defects by positional cloning. So how people do forward genetic screen is identify a defect in an organism, and you don't know why. You can take an animal, do some expose them to some mutagen, or you create some mutations in the genome, and then you see whether it is able to regenerate or not. And if it is not able to regenerate, what we know, I damaged some part of the genome, or some gene is defective now, but because of which it is not able to regenerate. But who will tell you what is that gene? So you have to identify, and that is called forward genetic screen. So you have to do a positional cloning. I will not go into the details of positional cloning. You need to have a lot of genome markers, etc. But you can eventually narrow down and identify which chromosome, which part of the DNA or which part of the genome has got this gene that is defective now, because of which the animal is not regenerating. The reverse genetic screen is other way around. You have a gene in hand and you don't know what is its function. You disrupt it, knock down, knock out or something, and then see what is it doing. That is other way, the backwards is called a reverse genetic screening. So lot of such research have done in mutagenic studies have shown that regeneration can be made compromised in zebrafish due to defects in, or due to induced defect in some genes. New advances in the high throughput genome, exome and transcriptome sequencing are likely to reboot the forward genetic approaches to study regeneration. Since we know the genome of the entire animal and even the sequencing of the genome of a mutant which you have in your hand is rather easy these days. Hence, identifying a defect is far easier. You don't have to typically go for the positional cloning. So this is a very interesting picture, that is the time-lapse imaging of the fin after amputation, that is done at around 27 degree. That is a temperature in which the zebrafish is housed. Normally the water temperature is around 27 degree. This is an uncut fin, and this is a 1 day after amputation. So this is the plane where it is cut, and it is called a DPA. 1 DPA means 1 day post amputation, and this is 3 DPA, this is 6 DPA, this is 12 DPA, and this is 20 DPA. 20 DPA and uncut look more or less the same. There is no difference. So here the whitish cell mass that what you are seeing is called blastema, and here there is barely any blastema. Here little bit blastema, there is more blastema. Here it is blastema is there, but the fin is already formed. Here there is no blastema, the fin is completely formed, and it is also restoring the same stripe pattern as you had. Slight difference is there here, but it is more or less the same in 3 weeks of time. And the same thing in a higher magnified level, what you are seeing in the B panel, the blastema at a higher magnification, you can see here. And you look at this panel C, what is happening? So this is homeostasis, that is steady state level in an uncut condition, and this is amputation at 0 day, and the wound closure will happen within few hours. And then what happens, there is a disorganization and dedifferentiation happens before 1 DPA, and in 1 DPA you have got mesenchymal proliferation and the blastema formation, gentle or slight quantity of blastema that you can see in 1 DPA. You need a high magnification, and then you have got a blastema outgrowth. There will be an expansion of the blastema, and also the actinotrichia formation between 1 and 3 days, and by around between 5 and 10 days, what you see, you will have the apical blastema maintenance. Extreme end, you need to still have to have the blastema between 5 and 10 days, and the rapid growth of the fin happens behind, that is between the blastema and the cut region. There is a formation of the differentiation of the fin, and then progressive redifferentiation. That means the blastema give rise to the fin tissue, and then it has to have the morphogenesis, that is morphologically the old structure has to be restored back. And by around 20 days, between 20 to 25 days of time, there is a termination of regeneration. That means you don't need blastema anymore, and the homeostasis is restored as you found in the uninjured condition. Those who are interested can read this paper what is cited here. So if you look closely, the zebrafish fin regeneration, the amputated fin ray is covered within the first several hours by the epidermis, and a regeneration blastema forms within 1 to 2 days. So soon after injury, there will be a blood clot that will be formed, and then there will be a migration of the cells from the surrounding adjacent side of the fin. If I can show here, if there is a cut here, the layer will be formed here by the movement of cells from here. So that it's acting almost like a blanket. And if this epidermal layer covering is not formed, due to some reason you stop it, you cannot get any blastema that is being formed. Or in other words, this epithelial layer that is formed from the migration of cells from the surrounding tissue is essential for the blastema to kickstart. So blastema take around 1 to 2 days. The blastema is a proliferative mass of morphologically similar cells formed through disorganization and distal migration of fibroblast and osteoblast or scleroblast proximal to the amputation plane. So two terminology you should know, proximal and distal. So proximal means something closer to the body. Distal means something away from the body. This is what you should get. If you have a fin, if I can show the previous picture once again. So this, because the animal is below this. So this is a proximal fin, this is the distal fin. So as with fin blastema in other classical regenerating system such as salamander's limb and the planarian's head, and the fin ray blastema is the major source of new structures. You see in salamander limb regeneration also, planaria's head regeneration also, you see the formation of blastema. Here in fin regeneration also, the blastema is must, and blastema does more or less similar job or similar function. So if you look closely, the anatomy of the blastema, because it is very important in multiple ways. First thing is, the anatomy tells you what are the tissue types that need to be formed. Say if the fin is made of muscle, neuron and bones. After regeneration, you cannot have just a group of fibroblasts. You need to have these structures formed precisely in the precise place. It should be an extension of the existing body part. You don't want a totally different pattern that is being formed. So understanding the anatomy is very important. If you see here, you can see there are 16 to 18 rays, and this is the proximal fin, this is the distal fin, and here is the amputation plane. And if you see, the blastema that is formed is basically an extension of the 16 to 18 rays you can see when it is being formed. And the higher magnification, when you look at it, you can see much more better structures. This brown colored one is the ray, and then this slight yellowish color one is the newly formed rays in the blastema, and it is a proximal and distal, and the distal most blastema do not have the ray because it is still growing. It is slowly getting into or walking into that zone. And if you look here, what you are seeing here is you can do sections in multiple places. Like you can have a proximal, distal and distal most. That is, this is a cross section from here, proximal, and then you have got a distal and the distal most. So like I told you, distal most do not have any fin rays. So this is a cross section. So when you look in this cross section, see, you can see this proximal one has got something called hemirays, and these rays are something like a bracket, It is not a cylinder, so it's like a bracket, so that it will allow some flexing. So that is what you are seeing here. You have got hemirays that is located, and what is in this green color is the actinotrachea, and the osteoblasts are slight brownish in color. Bone matrix is deep brownish in color, and epidermis, which is the skin epidermis, which is gray in color, and the red color circles are the blood vessels. So you can see in the proximal, you have got very thick bony matrix of the fin rays, but still the hemirays are seen nicely. So ray, between ray is interray, and each ray is made of two bony plates. That is, each plate, half plate is called hemirays. And proximal have got much thicker bony plate, and distal one has got the rays, but it is not that much thicker, and distal most has only blastema, but it has got actinotrachea is there, epidermis is there. All it is lacking is the blood vessels and also the thick ray or the calcium or the bony structures are missing. And if you look here, you can also see the lateral view of the distal most blastema, distal blastema and the proximal blastema, and this is the stump. Stump basically means the remaining part of the body. So the regenerating stump of the caudal zebrafish fin 3 days post amputation is what is been shown here. This blastema formed is almost 3 days post amputation, and that is you can recognize by this dashed line, and rest of them are very easily understandable when you take a section and look under the microscope, and you can stain them under the microscope so that you can get a better view of this whole structure. So organ regeneration is basically means the restoration of cellular and physiological normalcy of an organ. As you can see here, if there is a damage that occurred, like this is the structure which normally present, and this are developing, and this extreme end is the growing cone or the growing the blastema cells. They are trying to mimic that of this stump. So this is what you should keep in your mind, and this light brownish color is retaining this original structure, but it is not complete yet because it will take around 21 days. So that is what this organ regeneration, restoration of cellular and physiological normalcy of an organism. Just having a structure similar is not going to help. If you lost your nail, you don't want some other structure, you want nail only, which should look exactly like the nail what you had before damage. So if you look fin regeneration closely, zebrafish fins are complex appendages that quickly and reliably regenerate after amputation, restoring both size and shape. So the key regenerative units are the many rays of the dermal bone, which are segmented and lined by osteoblast. The word osteo, when you hear itself, you should know it is a bony structure. Rays are cylindrical and hollowed. Like I told you, they are like this. They are cylindrical, but they are not touching each other. It's like a two bracket in the cross section. You can see like that. The two concave hemirays surrounding an inner mesenchymal tissue that is innervated, vascularized, and comprised primarily of fibroblast. Fibroblast, we know they are sticky tissues and they are present. They do the blanketing function, and there is a innervation is there because the bony structures also should be mobile, and they need to have a muscular structure, and all these structures together give rise to a functional fin. An amputated fin ray is covered within the first several hours by the epidermis, which I already told you, and within 1 or 2 days a regeneration blastema has to be formed. So either you block the epidermis formation or you block the blastema formation, just forget about getting the proper regeneration. So the blastema is a proliferative mass of morphologically similar cells. We can compare them to that of the blastomere cells or the stem cells that is there in a developing embryo. Something similar, although they are not identical, something similar, because it can give rise to all cell types present in a normal fin. Although it is the blastema is a congregation of different types of cells, they can differentiate in the right place. That is the beauty of this regeneration. And they are morphologically similar cells formed through disorganization and distal migration of fibroblast and osteoblast or scleroblast proximal to the amputation plane. So blastema formation is only one step in zebrafish fin regeneration. That means it is looking in the right direction. Just because there is a blastema, we cannot be sure that fin will be formed. That is the beginning step or the first successful step. And the fins must then grow to its appropriate size, and the regenerative outgrowth occurs by two major processes. One is maintenance of a proliferative compartment at the distal end. That means in the previous picture we saw there is a stump, the proximal, distal and the distal most. And the distal most part should have blastema as it is. From the very beginning, whatever blastema formed, it should be there in the distal most plane. And the differentiation of the more proximal cells, they keep differentiating. If I can quickly show this once again, when the blastema is being formed, I will show here, blastema is being formed, this area, there will be, this is the proximal part, it will be differentiating. As you can see here, there is even pigment cells etc. are formed, whereas here there is no pigment and they are still looking whitish. So it is not waiting for the full fin to form. Even before this is reached, even the cells that is proximal in this boundary, stump and the proximal fin is now differentiating. And it is just walking into the extreme tip. So distal end of the fin retain this blastema, and the differentiation happens in the more proximal ends. So if you look fin regeneration, there are so many growth factors that comes into picture. The proliferative compartment is maintained by signaling interactions between the mesenchyme and the distal epidermis. So mesenchyme, like I discussed in the previous classes also, in the mesenchymal epithelial to mesenchymal, mesenchymal to epithelial transition, the mesenchyme is something which has the ability to migrate. This is what you should keep in your mind. A normal epithelial characteristics, despite having all the abilities, it is not free to move. So the mesenchyme has the ability to move. So If you want to have the blastema to be formed after an amputation, the formation of mesenchymal cells is a must. The proliferative compartment is maintained by signaling interactions between the mesenchyme and the basal epidermis. So these two has to cross talk. Then only you will know how much proliferating cell has to be accompanied, or how many in the blastema and how many cells has to be differentiating. In addition to regulating blastema formation, one molecule called retinoic acid. Retinoic acid, all of you would be familiar with. Retinoic acid, another common name is vitamin A. And fibroblast growth factor, FGF, mainly FGF2 plays a major role, whereas other FGFs also contributes. And canonical Wnt signaling positively regulate the blastomal proliferation and outgrowth. These signaling event, retinoic acid, FGF and the Wnt signaling contributes to the positive outward growth. The Wnt signaling pathways are a group of signal transduction pathways made of proteins that pass signals from outside of the cell through the cell surface receptor to inside the cell. So the ligand of Wnt signaling, they are called Wnt, and the receptor of Wnt signaling, they are called frizzled. The moment Wnt bind onto the frizzled, it will create an environment inside the cell, because of which the beta-catenin will be stabilized. The beta-catenin is normally degraded. Every cell has got beta-catenin, but the excess beta-catenin will be degraded. But Wnt signaling makes sure that the beta-catenin is not degraded. If beta-catenin is not degraded, it will go into the nucleus and create lot of pro-proliferative gene expression events, which is needed for the blastema formation. So that is the typical canonical Wnt signaling. 3 Wnt signaling pathways have been characterized. 3 Wnt different types of action. One is canonical Wnt signaling pathway, and the non-canonical planar polarity pathway, and the non-canonical Wnt calcium pathway. So the Wnt ligand can make the vicinity to respond in 3 different angles. All 3 Wnt signaling pathways are activated by binding of a Wnt protein ligand to the frizzled family receptor, which passes the biological signal to the protein disheveled inside the cell. Disheveled is part of the destruction complex, which normally degrades the beta-catenin. Once the disheveled is hijacked by this frizzled, then the degradation of beta-catenin is prevented, hence it will go to the nucleus in a typical canonical Wnt signaling event. So you can see here, the canonical Wnt pathway leads to the regulation of gene transcription. Like I told you, many genes, even MYC is there, lot of many cyclins are there. These are all turned on because of the Wnt signaling. Remember, Wnt signaling is important. We have seen in planaria regeneration also, Wnt signaling is essential for lot of biological pathway. And even if you have got a scratch wound, even if you have a mosquito or ant bite, the healing involves Wnt signaling. And many times the cancer also formed because of Wnt signaling. The non-canonical planar cell polarity pathway regulates the cytoskeleton. So the canonical Wnt signaling pathway turns on the gene expression events, whereas another non-canonical planar cell polarity pathway of Wnt signaling regulates the cytoskeleton, that is responsible for the shape of the cell. The newly formed cells must have a particular shape. The mesenchyme should have a spindle shape so that they can migrate. But once it is lost its mesenchyme property as a part of differentiation, it is retaining its epithelial property, then it will settle down and the morphology changes. So the regulation of the cytoskeleton is important, because cytoskeleton decides what should be the shape of the cell. That is responsible for the shape of the cell. That is also governed by this Wnt signaling. And the non-canonical Wnt calcium pathway that can regulate the calcium inside the cell. So which is also important for various signaling events. So you can see here, this is 1 day after amputation, the control, and 7 days after amputation, normal fin is formed. Whereas Wnt signaling, when you inhibit it, you don't get it. It's even after 7 days, it looks just like 1 day itself. That means if you block the Wnt signaling, you do not get normal regeneration. We study more about fin much later class, and we will learn about different aspects of regeneration in the next class. Thank you.